All right, hi everyone. My name is Carl Dargitz, and I am a R&D scientist here with Thermo Fisher Scientific, working within cell biology. And today I'm going to talk a little bit about embryonic stem cell FBS that's been qualified, which is a specialty serum product. And before I get really into that, I just wanted to encourage everyone, if you're checking this talk out, to check out some of the other content we have going on all throughout this time period, all day, and there's some really good and beneficial information out there. So I hope that if you're exploring this, you'll also look into some of the other great things that we have on deck as well. So to first talk a little bit about embryonic stem cell culture in general. So this field has evolved quite a bit since the very beginning. And we're at the point now, especially within human stem cells, where most of the culture systems are actually fully defined medias. However, traditionally, mouse embryonic stem cells were actually cultured using fetal bovine serum, or FBS. And this was also true in some cases with human cells. Because of this fact, and because FBS is not a fully defined additive, it really necessitates very stringent testing. And the main thing that needs to be ensured when that testing is done is that it's able to support undifferentiated growth of embryonic stem cells. And the image on the right that you can see there shows what you would be looking for if you did have some mouse embryonic stem cells that were grown in media containing qualified FBS. And I'll get into the details of that morphology and what they should look like a little more. But before I do that, I just wanted to highlight some of the different options and methods that are currently used in order to qualify FBS as embryonic stem cell grade. One of those options is colony-forming assays. Another is immunostaining. And then there's also options that allow you to quantify the pluripotency gene expression, either through flow cytometry or qPCR. So we considered all these options when we designed the assay for our FBS, and ended up settling on a version of a colony-forming assay for a few different reasons that I'll get into on the next slide. So this slide gives you a better idea of exactly what I'm talking about when I talk about our colony-forming assay. So it begins with thawing cells, specifically the PRX129X1 murine embryonic stem cell line. And it looks at both plating efficiency and pluripotency maintenance. So after these cells are thawed, they're allowed to grow for a few days in media containing your FBS that we're testing. And then we also have a positive and negative control. So the negative control being a condition that does not have LIF, and the positive being one that does contain LIF. So LIF is leukemic inhibitory factor, and that is an important additive that allows the stem cells to grow the way that we would expect them to. And again, I'll mention that again later. But as you can see in the left panel, after we allow the cells to grow, we perform alkaline phosphatase staining, and then image those wells and then count them. So as you can see, the difference between the LIF and no LIF conditions are quite significant. What we look for when we're looking for AP positive or alkaline phosphatase positive colonies is a very bright red stain without any fuzziness or ill-defined borders. So these are very tightly packed, bright colonies, as you can see in the upper image of the left quadrant. By the same token, the condition without LIF has very few of these and is much lighter in color, more fuzzy and spread out. And so after we perform that count, we do a stain with methylene blue, which is a general cellular marker. And this allows us to calculate the total number of colonies within each well. once we have that number, once we image and count those colonies, we're able to then divide the total number of alkaline phosphatase positive colonies by the total number of colonies, which gives us a percentage of alkaline phosphatase positive colonies, or in other words, a percentage of pluripotent colonies within the well. This really is a useful tool because it allows us to, as I said, quantify the number of pluripotent colonies, which is, again, the most important measure that you can have. As I mentioned, we looked at other options for this assay, but without a full genome-wide transcriptome analysis, you wouldn't be able to get the same thing you're getting with just looking at the number of positive AP colonies as a percentage. So looking at the graph on the right, we also looked at the consistency of this assay by looking at 3 different users performing that assay and how their results compared. As you can see, all 3 of the users resulted in very similar numbers of AP positive and colonies as a percentage. And so this gives us a high level of confidence in our FBS's, in our assay's ability to be consistent and without high amount of variability. So I mentioned the cell line that we're using for this assay, and you can see the full name of it there in the first bullet point, but I'll refer to it as PRX129X1, and this is a murine embryonic stem cell line that we get from Primogenics. And so after this cell line was grown in media containing our FBS, we wanted to make sure that it was behaving the way that you would expect and it wasn't having any issues. So the first thing that we wanted to look at was the karyotype of our cell line. It's important that for stem cells especially that this is a normal karyotype. And so that's what you see in the bottom left-hand corner is the karyotype analysis from a traditional G-banding method. And you can see that it is came back normal, which again is important. A lot of different stem cell lines, whether it be human or mouse, tend to acquire karyotypic abnormalities when they're grown, and so it's important to monitor that and make sure that there are no issues there. Second thing we looked at was LIF responsiveness. I mentioned LIF earlier, that's the leukemic inhibitory factor, which is an important addition to media for mouse embryonic stem cells and allows them to maintain pluripotency. So if a mouse embryonic stem cell line is LIF responsive, it means that with LIF in the media, it's able to maintain pluripotency. And you can see in the graph that the positive LIF condition does have a high percentage of pluripotent colonies as calculated by the alkaline phosphatase positive colonies divided by the total number of colonies. And by the same token, the negative the LIF negative condition has very few positive pluripotent colonies. The other things that we took a look at were the expression of pluripotency markers by both immunostaining and dyes, and also just morphology. So in the quadrant of images on the right-hand side of this slide, the upper left is just a phase contrast image at 5x magnification. And these cells really exhibit the hallmark traits of pluripotent stem cells, including a sharp refractive edge, tightly packed colonies with the cells growing very close together, and high nucleus to cytoplasm ratio. To the right of that is the live AP stain, which is a dye that transiently diffuses out of the cell over time and allows for use of these cells in later applications. It's not a terminal endpoint assay. And as we mentioned earlier, alkaline phosphatase is a marker of stem cells. The bottom 2 panels show OCT4 and SOX2 immunostaining, which both are important transcription factors that are pluripotency markers. And you can see high levels of expression for both of those in those 2 images. I also wanted to highlight the fact that ESL qualified FBS is not only useful within your traditional standard culture practices, it's also able to be used in more sensitive assays, such as reprogramming. So this slide shows you the results of a reprogramming experiment that we performed with both human and mouse fibroblasts using Cytotune 2.0 reprogramming kit. And what that is, that's our Sendai virus-based reprogramming kit. And so you can see the workflow there across the top. At day 0, we transduced the fibroblasts for both the human and mouse cells using Cytotune 2.0. And then at day 7, these were replated onto a feeder layer of cells. And the media at that point was changed from the fibroblast medium, which is DMEM with 10% ES-qualified FBS, to an ESC medium, which is slightly different whether it's human or mouse. For the human, we have DMEM with 15% ES-qualified FBS and FGF, whereas for the mouse, we have the same basic composition, DMEM with 15% ES-qualified FBS, with an additional mouse LIF instead of FGF. And after that whole process, you can see the colonies that were generated. These 2 wells are showing an alkaline phosphatase stain again to identify the pluripotent colonies. And you can see that a significant number of colonies were generated for both human and mouse cells. And these were, pluripotent colonies that were able to be picked and turned into usable cell lines later on down the road. So again, the point of the slide being that this FBS is able to be used in more sensitive assays and processes such as reprogramming, not just your standard routine culture. So the next thing we wanted to take a look at was how our FBS stacked up against the competition. So we did this by taking our same cell line that we use for our qualification assay, thawing it, and then passaging that into 5 different medias that were the same except for the FBS that it was supplemented with, and that included FBS, ES-qualified FBS from Thermo Fisher Scientific, as well as 4 competitors. Then, now that the cells had been separated into the medias with different FBS, they were grown for 3 passages, and finally analyzed for population doublings, morphology, and pluripotency markers. So I'll start by highlighting the population doublings. And over the course of the 3 passages, we saw that all 5 conditions, all 5 different FBSs from the 4 different companies and Thermo Fisher showed similar doubling times, which was between about 6.5 and 8 hours. As far as morphology goes, the cells that were cultured in embryonic stem cell qualified FBS from Thermo Fisher actually had a relatively higher degree of homogeneous colonies with that classic morphology of undifferentiated stem cells that I mentioned earlier, which has that tight, tight border that's very refractive, cells that are grow very close together, and a high nucleus to cytoplasm ratio. And we saw that more consistently in the Thermo Fisher media. The final measure that we looked at for the comparison of the different FBS qualified, embryonic stem cell qualified FBS lots from different companies was pluripotency marker expression. We did this by using flow cytometry. So on the far left of the screen, you can, the far left unstained control that's in black, you can see we use that to set our gate for our SSEA1 marker, which is what we used in order to evaluate the pluripotency marker expression. And this is a classic positive pluripotency marker for mouse embryonic stem cells. We also had a positive control, which is what we ran initially, right after right before the cells were split into their different media and FBS conditions. And as you can see, that is largely positive for SSEA1. When you look at the different conditions that we tested, you can actually see that Thermo Fisher Scientific FBS really recapitulated the positive control very closely. In other words, over the 3 passages, the percentage of positive, SSEA1 positive cells didn't change very much at all, stayed very consistent, whereas for all 4 of the competitor FBSs, that number dropped a little bit. So now that you've kind of seen the design of the assay, which allows you to qualify, that allows us to qualify embryonic stem cell FBS, and how well it does against, other companies' FBS as well, I just wanted to highlight the options that are available for our FBS in terms of both the origin of the FBS and the size of the container. So we have 100 ml, 500 ml, and then also the one-shot format, which I'll talk a little bit more about. As far as origin goes, you can get the FBS from USDA-approved regions, as well as U.S. origin and New Zealand origin. So there's a lot of options in terms of not only the origin, but also the size you might need depending on how much FBS you're going through in your workflows in your lab. So to expand a little bit more on the one-shot ESL FBS, this is available in the ESL qualified version, and the benefit here is that it's aliquot free, so you're not forcing your FBS to go through an extra freeze-thaw cycle, which is very beneficial, because if you're not using a whole 500 ml bottle and you need to aliquot it, you're putting your FBS through an extra freeze-thaw cycle, which can be problematic. If you're using a ton, this may not be beneficial for you, but if you're using small amounts at one time, then this can be very helpful. This has also been improved on from the first generation by making a wider mouth so that all pipette tips can fit in, and it also fits nicely in a 30 millimeter test tube rack. I also want to mention that there are free samples available on the Thermo Fisher Scientific website. As you can see from the screen cap, there's just a little button there, and it's just a few lines to fill out before you're able to obtain a free sample. So, if that's something you're looking at testing, this would be a great opportunity to go check that out and get what you need. So in summary, the advanced assay that we used to qualify our ES-qualified FBS was designed to sustain undifferentiated embryonic stem cells while maintaining karyotype integrity, lift responsiveness, as well as pluripotency markers. And it has proven applications in iPSC generation and PSC culture, including reprogramming, pluripotency marker maintenance, as well as pluripotent stem cell morphology maintenance. We also saw that it had superior performance over competing products with regard to morphology and especially maintenance of pluripotency. And there is a newly designed aliquot-free one-shot version available now, and you're able to get free samples of that on our website. So, thank you.